Next presentation we have is from Rebeccah Ayako from Kenyatta University in Kenya. And she'll be talking about the concomitant infection of Leishmania, donovani, and Plasmodium berghei, and how that reduces disease severity in BALB mice. Ayako, please take it away.
Hello everyone, my name is Rebeccah Ayako from Kenyatta University and Institute of Climate Research. I'm quite honored to be part of this team where I get to share my data on how disease severity is reduced during Leishmania donovani and Plasmodium berghei co-infection in BALB mice. Leishmaniasis is a neglected tropical disease which is endemic in 88 countries. It is transmitted by phlebotomin female sandflies. 350 million people are at risk of infection, with 500,000 cases reported yearly. Clinical forms of leishmaniasis include cutaneous leishmaniasis, mucocutaneous leishmaniasis, post-caloza-dermal leishmaniasis, which is a complicated form of visceral leishmaniasis that is 90% fatal if left untreated. Reported cases of visceral leishmaniasis vary between 50 to 90,000 cases annually. Malaria, on the other hand, is transmitted by female Anopheles mosquitoes. In 2019, WHO reported 229 million cases with 409 deaths globally as a result of malaria. Visceral leishmaniasis and malaria continue to be the principal causes of morbidity and mortality associated with parasitic infections. So with these infections presenting similar clinical features such as fever, anemia, and weight loss, and taking into consideration inadequacy of qualified medical attention and laboratory facilities, it is unclear whether the right disease is diagnosed. And also, it is unknown whether these infections could impede with both serological and parasitological disease diagnosis and both symptoms. So in the map below, you can see regions of endemicity, parts of East Africa, South Asia, and South America. So it is with this co-infection areas that prompted me to characterize clinical features such as body weight, parasite burden, and pathological changes in the spleen, liver, tissues, and survivorship, together with immunological features such as immunoglobulin gamma levels present in mice which are co-infected with Leishmania donovani and Plasmodium berghei. So I had 82 mice in general, which were in general, which I had divided them into 4 groups. I had the experimental group, which contained Leishmania donovani and Plasmodium berghei parasites together in the same mice. And I had the positive controls, which were Leishmania donovani group, Plasmodium berghei group, and the naive group, which was not inoculated. So for Leishmania donovani inoculation, the mice contained 1 million promastigotes, while Plasmodium berghei groups contained 20 million infected erythrocytes. So in the co-infected group, mice were inoculated with Plasmodium berghei 60 days post L. donovani inoculation. Body weight was determined on a daily basis post co-infection up to the 10th day, well, that was my termination period, while parastemia was carried out on a daily basis from the 4th day post co-infection to the termination period. Mice were sacrificed on day 0, 4, 6, up to the 10th day to harvest serum, which was from blood, for IgG quantification using ELISA assays. At the same time, spleen was harvested from Leishmania infected mice from both the single and the dual to quantify parasite load, which was in the spleen using splenic impression smears. On the 9th day post co-infection, 1 mouse from each group was sacrificed and spleen, liver, and brain tissues harvested for determining histological analysis. 4 mice from each group was left untouched, for which survival rate was determined. So when you look at the results, body weight in dual and singly infected mice groups, they decreased significantly between the 0, this was 60 days post pre-co-infection. So there was significant decrease in both the single and the dual infected groups. However, when you compare these differences between the single and the dual infected groups, it was not that significant. When we look at the Leishmania donovani parasite burden, in the dual infected mice group, there was a reduction. That means there was some sort of protective immunity during the first phase of co-infection, and then before it started increasing slowly and gradually, and results shows that there were significant differences between the day 0 and day 10 post co-infection, and the same was observed in the Leishmania only infected mice group. And when you compare this single and the dual infected mice groups, there was significant differences in Leishmania donovani parasite load. Looking at Plasmodium berghei parasitemia, there was a delay here from day 4 and day 5 post-co-infection. There was a delay in the onset of parasitemia in dual and in the dual infected mice group before starting to increase slowly up to the 10th day. And when you compare the single and the dual infected mice group, there was significant differences in the parasitemia. When you look at the specific IgG responses targeting Leishmania donovani, we can see that there was an increase in IgG responses in both the dual and the single infected mice group between day 0 and the 10th day of co-infection. But when you compare these two groups, there was no significant differences. And there was positive relationship between parasite load and Leishmania donovani specific IgG responses, meaning that an increase in parasite load resulted to an increase in specific IgG responses and vice versa. When we look at parasitemia levels, the case was quite different. So there was significant differences between the single and the dual infected mice group in terms of IgG responses. And also there was a significant increase in IgG responses between day 0 and day 10 of co-infection period. And there was a moderate positive correlation between the single and the dual infected group when you compare in terms of parasitemia and IgG responses, meaning that an increase in parasitemia resulted to an increase in IgG responses and vice versa. When we look at histopathological changes in the spleen, result shows that there was less disorganization. So group box A points to Leishmania, the dual infected group. Box B is the Leishmania only infected group. Box C shows the Plasmodium berghei infected group, and box D is the naive group. So here we can see less disorganization of granulomas as compared to here. And then when you look at the enlargement of the red and the white pulps, you can see here it was less pronounced as compared to the co-infected, as compared to the singly infected Leishmania group. When we look at the liver, we can see here granulomas pointed by the red arrows here. And then when you look at in the dual infected group, you can see less granulomas which are not quite defined. And then there's congestion of blood vessels present in the Plasmodium berghei infected group pointed by the brown arrow. And when you look at now the dual infected group in general, you can see hyaline deposits, these small dots here pointed by the white arrows. And then there's necrosis and tissue injury. You can see this place is somehow highlighted, it's whitish, meaning there's a lot of tissue damage occurring here. I focused to look at the brain to see if there was any form of cerebral malaria observed during co-infection, and the results were quite amazing. So when you look at the Plasmodium berghei only infected mice group, we can see microhemorrhages here, and we can see the massive sequestration of leukocytes here and also here. But when we look at the dual infected mice group, we can see few leukocytes present here, meaning that parasites were able to cross the blood-brain barrier, but not as pronounced as the malaria-infected group. So when I was looking at the survivorship curve, the Leishmania donovani and naive mice never succumbed to any infection during the 20 days of infection. So here, it's important to note that there was a region of overlap between the blue and the black line. So no mice died in the Leishmania donovani and naive mice group. But when you look at the co-infected group here, the green, the last mouse died on the 13th day, while Plasmodium berghei, last mouse died on the 9th day. So there was high mortality rate observed during Plasmodium berghei infection. So I'm able to say that an increase in parasite burden and survivorship resulted to a decrease in body weight. And body weight reduction is normally attributed to, in terms of malaria, it's attributed to anemia, and in general, loss of appetite. And an increase in parasite burden resulted to an increase in specific IgG responses in both dual and single groups, regardless of the mode of infection, either it was Leishmania infection, Plasmodium infection, or dual infections. And it is important to note that specific Leishmania donovani specific IgG responses are not protective in any way, but they're a marker of disease progression. That's a very key during diagnosis. And during infection, there was reduced granulomas, microhemorrhages, and enlargement of the pulps, which is a positive indication. However, when you looked at the liver, the case was quite different, since there's a lot of infection going on on malaria in the liver, and also hepatomegaly observed in Leishmania patients. The case is quite different. You could see hyaline deposits, which are a marker of disease progression, and at the same time, you could see lymphocytosis and necrosis observed. So I'm able to conclude that Leishmania donovani and Plasmodium berghei co-infection is able to reduce disease severity by 35.3% in visceral leishmaniasis and 21% in malaria. And when you look at IgG antibodies in dual infected mice, it was 4% higher in Leishmania donovani and 5.99% higher in Plasmodium berghei. So based on these findings, I am able to recommend policymakers to implement early detection as a new way of diagnosis by use of less invasive methods such as polymerase chain reaction that is able to reduce mortality and morbidity experienced in regions of co-endemicity. So that brings me to the end of my presentation. Thank you for listening.
Well, thank you so much for your presentation, Rebeccah. It's a very, very interesting talk. We have a couple of questions from Dr. Eleanor Fish.
Thank you. Yes, that was a great talk, Ayako. Thank you very much. So I guess one of my first question is, cognizant that L. donovani is a relatively, mild infection in your mice. What was the basis of the selection of the various infective doses for the two that you used? How did you come up with those doses?
Okay, based on what other people have done in characterizing Leishmania, characterizing animal models such as BALB mice for L. donovani inoculation, the best dose was 1 million promastigotes, and for the people who have done in treating Plasmodium berghei in mice, they've been able to quantify the pellet when you spin blood which contains infected RBCs, and you are able to know the percentage of parastemia, and then now 20 million infected RBCs is able to infect mice. But when you use less numbers, it's able to just flush the whole infection, and there's no infection present. So that made me conclude to using 20 million infective RBCs.
Okay, but with the L. donovani, what was the dose you used there? That was 1 million.
1 million promastigotes.
Yeah. Okay. All right. And beyond, so your survival studies perhaps you weren't surprised by them, because you knew you had better survival with the donovani. So beyond IgG, what, if your next steps, what would you actually look at?
Okay, so I'm quite fascinated about learning on the protective immunity, which we could see from the mice. And since malaria infection is quite common, there's production of interferon gamma, which in terms of infection of Leishmania donovani, it's more protective. And then I will very much want to go further in seeing pro-inflammatory and anti-inflammatory responses together with CD4 and CD8 cells responses in terms of co-infection, which is very, very important.
Thanks very much. Thank you.
Okay.
Great. Rebeccah, I have 3 questions in the Q&A from our audience. The first one being, can the different species of Leishmania be morphologically distinguished? If not, how do we distinguish them before infecting them into the mirrored model?
Okay, it's not easy to distinguish them, but using polymerase chain reaction, or molecular diagnosis, you can able to distinguish the different strains present. Yeah, and also you can look when you can use symptoms, like visceral leishmaniasis normally causes hepatomegaly and splenomegaly, and the other strains such as Leishmania major can cause cutaneous leishmaniasis. So you can able to see patches around the skin where there's an infection. So that can also be used, but the most common way is by using molecular diagnosis.
Molecular death. Okay, that makes sense.
Yeah.
Okay.
Okay.
And then another attendee had asked a question: how can we relate this results with Plasmodium species who infect humans? I suppose I'm assuming the ones that affect the mouse models if you have there.
Yeah, kindly repeat that again.
The question was, how can we relate this results with Plasmodium species that infect humans?
Okay, so the Plasmodium species that infect humans is Plasmodium falciparum. And for me selecting Plasmodium berghei, it is the one which is the clinical features present and the immunological reaction is somehow the same as observed in human infection. So from my data, we are able to model now what happens in real life situations in regions of co-endemicity.
Very interesting, very interesting. So another question was the parasite burden the only reason for increase or decrease in IgG response, or could there be another reason as well?
How parasite burden resulted to an increase of IgG responses?
Yeah, so I think they're trying to distinguish whether that may be the only reason for this increase in IgG response, or perhaps is there another reason rather than just the amount of parasites in it that can increase that?
So the number of parasites results to an increase in IgG responses. So you need to know that these mice were they are inbred, so there's no other infection that could lead to IgG responses. So it's basically the parasites which resulted to IgG responses. Okay.
Fantastic, Rebecca. Thank you so much for answering our questions.
If I can ask also a question,
okay.
Rebecca, this was a really wonderful talk, very interesting. And I'm always curious about when you have this sort of co-infection of the pathogens. One thing I'm particularly curious about, and I think there are a lot of interesting tools these days to look at this in mice, is what would happen if you have some of these more low-grade chronic bacterial infections that are there at the same time as a parasite in this case. So to give a more specific example, there are some strains of